Okay, so I didn't realize that was going to be so long, so I decided to split it into two parts. So we'll do the single genes and then the multiple genes in two separate videos. So this is a continuation of the genetics problem set, uh, part two, which will cover questions 12 through um, 18, which is plenty of practice problems for this. Okay, so I think we're on number 12, which is position of the flower on the stem of a garden pea is governed by a pair of alleles. Flowers growing in the axils, the upper angle of the stem, are produced by the action of a dominant allele T, big T, while those growing at the tip of the stem are produced by the recessive allele little t. Okay, so flowers can be colored or white, and colored is big C, white is little c. So we should write this down. So axial, which means in the side of the plant, like this. This is axial, and if the so here the flower is, or the flower could be on the top, and this would be terminal. Okay, so for the axial, it's big T, which is dominant. Little t is terminal. And then big C is colored. Little c is white, or the absence of color, the default color. So now we're going to get our crosses. We have a big T, big C, little c, female. Because we're doing a dihybrid cross, and it tells you each of these plants are dihybrid. It doesn't say anything about the sex. I'm just putting that in there. Dihybrid plant, color flowers, is crossed to a pure strain of the same phenotype. So in this case, we have big T, big C, big C. We can just make this the male. It's fine. So in the F1 generation, we can do a really easy Punnett square because there's only one possible gamete for this one, which is big T, right? Either this one or this one. And big C, either this one or this one. So we know that. And then this one could be big T, big C, big T, little c, little t, big C, or little t, little c. So we just do the cross, big T, big C, big C. Big T, big T, big C, little c, big T, little t, big C, or big T, little t, big C, little c. So here are the genotype ratios. Are one of these, big T, big C, big C. One of these, big T, big C, little c. One of these, big T, little t, big C, and then big T, big C, little c. So it's a 1 to 1 to 1 to 1 genotype ratio. And then for phenotype, well, this is going to be axial, and colored, axial, colored, axial, colored, and axial colored. So 100% doesn't. One of the answers I put it was purple, but it's fine. Any color is due. It's just colored. All right. So that's number 12. I think. Yeah. Okay. So question 13. We have W is white. Big W. Little w is yellow. Big S is disc. That's the shape. And little s is sphere. So it says summer squash, big W is dominant. That's white fruit. Yellow fruit is recessive, which is little w, which is what we read up there. Dominant allele, S produces disc-shaped fruit. Recessive allele, little s, produces sphere-shaped fruit. So we have the cross that it's giving us is homozygous white disc plant. So big W, big S. Cross of the homozygous yellow sphere plant, which is little w, little s. The F1s are all white and disc-shaped dihybrids of the genotype big W, little w, big S, little s. Okay, so we would expect that because this can only pass on a big W. This can only pass on a big S. This one can only pass on a little w. This one can only pass on a little s. So big W, little w, big S, little s. This would all be the F1 offspring. The two F1s are allowed to mate at random. What would be the phenotypic ratio expected in the F2 generation? All right, so we're just going to have brothers and sisters from plants mating. And so this is what we call a dihybrid cross. It's a straight up dihybrid cross. I don't need to do the whole cross out, but we could. I'll do it over here. So we have the gametes that you have to pass on one trait of each. So the gametes would be big W, big S, big W, little s. I might erase this just to get some space in here. Then little w, big S, big S, and then little w, little s. Same thing from this parent, so I don't have to go through the whole thing again. And so you can do the crosses out, like this is going to be big W, big S, and so on and so forth. But I already know the ratios because I studied genetics and I watched the lecture in chapter 14, and I know that when we do this kind of cross, we always get a 9 to 3 to 3 to 1 ratio. And this is 9/16 is going to be both dominant, so it's going to be white and disc shape. This 3/16 is going to be 1 dominant, so this could be white, and then 1 recessive, so this is, what is it, sphere. This is going to be 3/16, which is going to be 1 recessive, so in this case we'll call this recessive yellow, since it has to be different than the one before, which is white. And then this would be disc, because it would be the dominant. And then this one would be both recessive. So yellow and disc shape. So the question B goes on, and then you can do that one in practice doing the crosses if you want. I'm not going to do that here because there's a whole ton of crosses that you can do and you can practice on for that one. But it just follows the same sort of pattern. So if you have any problems doing those crosses, let me know and I can help you out. Okay, so 14. Back to fruit flies. So remember, they have that weird nomenclature. So the fruit flies here, we have little e is ebony, E plus is gray, VG is vestigial, which means they have tiny wings. You can look it up if you want to see what those fruit flies look like, but they have basically useless wings. And then VG plus is normal. So we're going to do the cross. Let me just double check the question. So if dihybrid flies are crossed, so we got to do a dihybrid cross, and that we know is one of each. So we're going to do this. So we have E. This is the parental generation. So we have E plus E VG plus VG cross to same thing, E plus E VG plus VG. So we don't have to do the whole cross out. We know that we're going to get a 9 to 3 to 3 to 1 ratio. And so we would, again, these are out of 16. So we would expect 9/16 of these flies should be gray, normal wings, both dominant. 3/16 should be gray and vestigial. Then we would expect 3/16 because 1 dominant, 1 recessive. 3/16 should be ebony, recessive, and normal, dominant. And 1/16 of these should be ebony and vestigial, both recessive traits. So this is our 9 to 3 to 3 to 1 ratio that we get from normal Mendel stuff. So it says that we have 256 flies, so all we have to do is multiply each of them by the ratio. How many of the progeny are expected in each phenotypic class? So for each one of those, all we have to do is multiply this by 9/16. For the first one, and that gives us 144. 256 times 3/16 is 48. 256 times 3/16 would be 48. 256 times 1/16 would be 16. Total, just to check our math, that total adds up to 256. So that's our answer. All right, next one is 15. 15 says short hair in rabbits is governed by. This again. So big L again. It's a trick. Big L is short hair. Little L is long hair. Black hair results from dominant allele, so big B is black, so that one's good. And then next page. Little b is brown. So it says, what phenotype and genotype ratio is expected from cross heterozygous short and black? So heterozygous short, big L, little L. Black would be big B, little b. Homozygous short and black. Homozygous short, big L, big L. Homozygous black is big B, big B. And then you can do out the Punnett square. That's a pretty easy Punnett square because we know that this parent can only pass on big L and big B. And so that's the only part of the Punnett square. And then for this one, we have the classic big L, big B, big L, little b, little L, big B, and little L, little b. So we just do the cross, L, B, and L, B. So we're just going to make this big L, big L, big B, big B. This one would be big L, big L, big B, little b, big L, little L, big B, big B, big L, little L, big B, little b. Turns out that they're all short. They're all short because that's dominant. And they're all black because, again, that's dominant. So 100% short and black. And then the genotypic ratio is what we drew out here. And that's a 1 to 1 to 1 to 1. So 1/4, 1/4, 1/4, 1/4. Homozygous short and brown rabbits. So we can erase this. this doesn't crash. Homozygous short and brown rabbits. So homozygous short, homozygous brown. Just do the same thing. This one can only pass on big L or little b. And then we just do the Punnett square again. This one is big L, big B, big L, little b, little L, big B, little L, little b. And then you just do the crosses out. And you can see that I solved for that in the solutions. So I'm not going to write all these in. You can complete that, but you know, it's just the same thing. So we have for this one, big L, big L, big B, little b, and so on and so forth. And then what you'll find out is that you're going to get, well, I'll just do this one out, and then the next one I'll let you do on your own. So this one is short and black. This one is going to be short and brown. This one will be short and black. And this one is going to be short and brown. So 2 are short and brown, 2 are short and black. So 2 out of 4 short brown, 2 out of 4 short black. That ends up reducing to 1/2 and 1/2. So it's a 1-to-1 ratio. I'll let you guys do part C on your own. Okay. So number 16. Number 16 says, JAG information for the following is found in problem 15. What phenotypic ratio is expected among progeny from the crosses of big L, little L, big B, little b? So this is a dihybrid cross, so we know that that phenotypic ratio is going to be a 9-to-3 or 3-to-1. We don't even have to do the cross because we memorized the ratios like good genetic students. So that's part A. Part B, just do the Punnett square. You'll get 1/16 is that, and 1/16 is that. All right. You can also do so for part B it says true breeds, what's the percent homozygous? So homozygous. So we could add these together. Rule of addition: 1/16 plus 1/16 is 2/16, which is 1/8. You can also do this a shortcut way because when we do crosses, you can do them individually. So it would be like if we did this cross to this cross. And so for this cross, what percent would we expect to get out of that? And the answer is 1/4, right? Because it's a monohybrid cross. It's a 1 to 2 to 1 ratio. Would be homozygous dominant. And then when we did this cross, here, we'd have big B, little b, big B, little b. And so what percent will we expect to be this one? And the answer is 1/4. So all we have to do is multiply this one, and this one would be big L, multiply by that, and that gives us 1/16, which is the same answer we got here. So it's just kind of a shortcut. And in the end, you get the same answer, which is 1/8. I'll let you guys do part C on your own. If you have any questions, send me an email. All right, number 17. So number 17, list all the different gametes produced by the following individuals. So you can do this a couple ways, but you do it by the power of the number of alleles. So for example, we already know that if we have this, then that gives us two possible gametes, right? Big A or little a. The way that you figure this out is that you do it the number of alleles to the power of that. So there's two different alleles, big A, little a. That power is to the first because we're only talking about one specific gene. And so if we were going to, so 2 to the first is 2. If we had 2 like this, then it would be 2 to the second power. So that would be 2 times 2, which is 4. And we know that we have 4 possible combinations. We can have A, B, big A, big B, big A, little b, big B, or little a, little b. And if we did it three ways, then it would be, so if we have, then it would be 2 to the third, which would be 8 gametes, right? So we can also multiply this out as individual outcomes. This one can only produce 1, so that's 1. We multiply it by 1. This one can only produce 1, so we multiply that by 1. This one is like this, right? There's 2 different ones, and we can put it to that 1 power, so that gives us 2 outcomes. So that 1 times 1 times 2 is 2 gametes that we can have, and that those 2 gametes are A, big B, big C, and big A, big B, little c. Does that make sense? All right. So knowing that, do part B. Little a, big B. So this one is 1 outcome. This one's 2 outcomes. This one's 2 outcomes, right? And so we get 4 gametes, and those gametes would be little a, big B, big C, little a, big B, little c, little a, little b, big C, and little a, little b, little c. I'll let you guys do the other part C and D on your own. I'm sitting on the floor, so my legs are cramping up. All right, so number 18. Cloven-footed condition in swine is produced by the homozygous recessive genotype little m, little m. All right, so we do little m is cloven. Big M, or big M, little m is mule foot. Big B is white. Little b is black. So it says so again, they're messing with this B as being the dominant thing. So just got to pay attention to what we're writing down as dominant recessive, because that can get confusing. Okay, so the cross is a white mule-footed female. So the parental generation, the female is white, mule-footed. All right. So what are the genotypes for white and mule-footed? White could be big B, or big B, little b, right? So we have a whole bunch of combinations for the female. We'll just write them out. So we have big B, big M, big M. It could be big B, little b, big M, big M. It could be big B, big M, little m, or big B, little b, big M, little m. Any of those possibilities for the female. And we're going to cross that to a male. The male is easy because we're told that the male is black, which has to be little b, and cloven. So this male has to be little b, little m, little m. That just happened. Okay, so then we just have to do the crosses. So the question is, what is the most probable genotype of the sow? So what is the probable genotype of this female from the information that we've given, which are 26 offspring, all were white and mulefoot. So all white. Right. So this and mulefoot. So that. So we know that if we take two pure breeding so this is homozygous dominant. This one is homozygous recessive. The only one that can pass on is big B. Here, the only one this can pass on is little b. The only one this can pass on is big M. The only one this can pass on is little m. And so these would all be heterozygous for the dominant condition, which is what we would expect for that cross. So the most likely female for that outcome would be the homozygous dominant conditions. So the next one is the next litter produced 8 white mule-footed offspring and 1 white cloven-footed. So with the information that you already have, you already know, you should be able to go ahead and work out that problem on your own as well. And then again, if you have any problems figuring out, you can come and ask me. So that's basically how you do these genetics problems.